you've all got to confirm you're still willing to stay on. Great. So welcome everyone, and it's a real pleasure to introduce Petronis, who's from Toronto and has been working in epigenetics before it was fashionable. Very important work that he said in his biography that he sent around, that some of you will have seen, that 5 of his papers are worth reading. So today's talk is definitely is about one that I think is a brilliant paper, and would highly recommend in Nature Reviews Genetics. But we're going to get the personal presentation of this today, which I'm greatly looking forward to. Thanks, Art.
Thank you, George. So welcome everyone. I cannot see you. I hope you can see my slides. And so for the next 45 minutes or so, my plan is to kind of introduce this relatively new term of chronoepigenetics, and to share some ideas how chronoepigenetic strategies could help us in our search of etiological factors of disease. So, I usually start my talks with a kind of broader context, and looks like I cannot. Okay, I can advance the slides. So, in a broader context, so where epigenetics is coming from, and how is it positioned relatively to other groups of etiological factors. So as you all probably know, all human diseases, or nearly all human diseases, are to some extent heritable. And once we talk about heritability, we nearly automatically think about DNA sequence variation, about polymorphisms, mutations, and so on and so forth. So this is the genetic side of disease, and whatever cannot be explained by genetics is traditionally interpreted as of environmental origin. So the traditional current paradigm can be reduced to a simple statement that etiology of disease is related to genetic risk factors which interact with environmental hazards. But we have a third player here, and there are some features, especially in complex diseases, complex non-Mendelian diseases, which are hard to explain by genetic or environmental risk factors. And here comes the potential for epigenetics. And I have a list of them. I'm not going to discuss all of them, but a classical example is discordance of identical twins. So the traditional interpretation is that actually there's something happening in the environment that genetically identical organisms would exhibit phenotypic differences. But this mantra has been repeated many, many decades. And actually, we still don't have some solid evidence that there are some real concrete environmental factors which would account for discordance, for schizophrenia, or diabetes, or multiple sclerosis. The second on my list is the so-called delayed age of onset in complex diseases. So the point here is that actually the inherited risk factors, genetic risk factors, do not cause disease for extended periods of time. And then actually it takes like 20, 30, 40, 50 years for the first disease symptoms to show up. And I think that actually this is like one of the most universal features of complex diseases. And it's so universal and so evident that actually we are trying, we do not, like, we stopped noticing that. Like, we are not trying to explain it. We're not trying to understand why is it so that genetic risk factors are silent, or they are basically innocent. They are not pathogenic for decades, and then something happens and they become pathogenic. So this delayed age of onset is one of the aspects that I will be discussing in greater detail from the chronoepigenetic perspective. And there are numerous other things, numerous other features, numerous other facets of complex diseases where we can provide an epigenetic explanation. So if you're not very familiar with epigenetic mechanisms and epigenetics in general, here is a slide which summarizes the kind of mechanistic, the chemical side of epigenetics. So from the chemical point of view, epigenetics is related to attachments to DNA and histones. And if you can see my cursor, like some nucleotides named the cytosines, let's say if we're talking about human or mammalian genome, cytosines can be modified, can be methylated, can be hydroxymethylated. And the same principle applies to histones. And histones can be methylated, acetylated, phosphorylated, and so on and so forth. So these attachments, this is actually what epigenetics is about from the chemical point of view. There are numerous functions of epigenomes and epigenetic factors. And traditionally, there are kind of three groups of three kind of types of definitions, depending on the primary role. And epigenetics is usually related to development and differentiation. Epigenetics is also a mechanism of memory of somatic cells, and epigenetics is also playing a role in regulation of gene activity. So in this slide, we have three very straightforward examples of the importance of epigenetic factors. So in all cases, we have genetically identical organisms or tissues, and actually we see huge phenotypic variation, and which cannot be explained by genetic DNA sequence-based factors, which cannot be explained by environmental factors, but can be explained by epigenetic factors. So if we think about the epigenetic context of epigenetics in the context of disease, it's not so difficult actually to rationalize that deviations from epigenetic programs could be an epigenetic risk factor. And again, like I showed in a couple slides ago, that there are numerous features, numerous facets, which are in complex diseases, which are very consistent with epigenetic, putative epigenetic misregulation. So that was my kind of general introduction into epigenetics. Now the chronoepigenetics, you can understand from the title, chronos is a Greek word which means time. So from the temporal dimension, a temporal dimension is very important in epigenetics, and epigenetic factors change quite dramatically during development, during cell differentiation, and in aging. And you probably heard about epigenetic clock. So epigenetic modifications of could be a couple cytosines, could be several hundred cytosines, can estimate the chronological age with high precision. So this is like, I'm kind of already turning into this temporal dimension, into chronoepigenetics, which is kind of an umbrella term for various aspects, various epigenetic processes in the temporal dimension. So I kind of had the second point, so second facet, the late age of onset. Why is it so that genetic factors can be silent for extended periods of time? So we're primarily interested in the late age of onset of disease, but actually it would be very difficult for us to investigate all those epigenetic processes in disease context. So we selected a phenotype which is very well known, which also has the late age of onset, but actually would be very much more easy to investigate. And that was lactose intolerance, or adult onset lactase non-persistence, the full scientific term. So a little bit of a background here. All or nearly all infants perfectly tolerate lactose. Lactose is milk sugar and one of the main sources of energy in infancy. We know quite a bit about genetics of lactose intolerance. So this is the lactase gene, and the adjacent gene is called MCM6. So it has nothing to do with lactose metabolism or processing, but it contains an enhancer in intron 13 for the lactase gene. And there is a polymorphic site in the enhancer. So C allele carriers would become intolerant at some point in life, and T allele carriers, would it be like only one T allele out of two? Two alleles, so one T, one C, or both T's, those cases will be lactase persistent, so they would be able to consume milk. So here is a situation, like in infancy, all infants can consume large amounts of milk, processed lactose, no problem. But some individuals, 5, 10, 15, or 20 years later, would develop lactose intolerance. And the situation smells like, to some extent, is somewhat similar to what we have in disease. So lactose intolerance is not necessarily a disease, but actually it could be a disease if you're not careful enough. So we thought that actually we already know a lot about this trait. We already know which the genes that are involved, the regulatory elements that are involved. And we know what specific cell types involved, which is in this case enterocytes or gut epithelial cells. And actually the question is like, why is it so that this phenotype is changing over time? Why is it so that we see phenotypic transformation in some individuals? So again, like we used this, not because we used this, we tested this phenotype, not because we're interested in it, but we thought that actually it's a good example. It's a very convenient case for the proof of principle. So we performed quite comprehensive analysis of this gene. Actually, we scanned the entire chromosome. I'll not be going into details, but we investigated DNA modification. And let's say just one quick comment that this is chromosome 2, human chromosome 2 is 250 million base pairs. And we detected statistically significant signals actually around the lactase gene. So that was quite encouraging, and we did some additional things. But the bottom line is summarized in the slide, that actually in young age, both alleles exhibit similar epigenetic modifications. So red bubbles represent epigenetic factors. Let's say these are DNA modification signals or methylated cytosines. And this is the enhancer region in MCM6. I don't know if someone could give me a signal if you see my cursor, like should I be using it or I should not? Can someone tell me if you see my cursor? Yeah, I'm not getting any messages.
So yeah, I can see a cursor.
You can see, yeah, very good. Okay, thank you, George. Yeah, so we have two alleles, two alternative alleles. This is a persistence allele, this is intolerance allele. And actually at young age, we have evidence, indirect evidence, that actually modification status is very similar. But actually what happens over time, that epigenetic factors start accumulating and DNA modification density increases, and the proportion of methylated cytosines is increasing in the intolerant allele, on the intolerant allele. And actually this is the enhancer region, and it becomes heavily modified. So again, on this allele, and this allele is somehow protected. So the conclusions that we made are the following, that actually it's not the polymorphism itself that plays the direct role in the phenotypic outcomes. And the logic is very simple, that infant LCT, lactase gene mRNA is high, and doesn't matter what haplotype it is. So the SNP is probably not directly involved in LCT regulation, but the SNP is probably mediates the age-dependent accumulation of epigenetic modifications, which directly orchestrate gene activity. So it's some kind of, it's a mediator of age-dependent epigenetic factors. And once those epigenetic factors reach the critical mass, so I guess that actually chromatin gets very dense here, chromatin structure becomes very tight, and actually this enhancer is inactivated, and then the actual gene would stop producing mRNA, and basically there's no protein, and we see all those phenotypic outcomes that we see. So this is the point that actually like there's interaction between genetic factors and epigenetic factors, and all those things are gradually changing over time. That was the main conclusion that we made in the study. And we speculated around that time that actually like complex diseases, including psychiatric diseases, could be like multiple cases of such lactose intolerance. Of course, it's not a gut phenotype. It's not enterocytes that are involved. It probably happens in the brain, and it's more than one gene and probably multiple genes. But the key principle is such that actually there are predisposing genes, and those predisposing genes could be relatively innocent for extended periods of time, but actually those predisposing genes somehow contribute to age-dependent epigenetic changes, epigenetic dynamics, and at some point it reaches a critical mass, and here we go, we have psychosis, if we talk about psychiatric disease. So here is animation actually to illustrate what I just explained. So we have the genome from one chromosome 1 to X and Y, and actually we have predisposing haplotypes marked in yellow bars. And those red bubbles that you'll start seeing soon are the epigenetic factors, which are, to some extent are dysregulated, which are not normal, which basically are mediated by predisposing haplotypes, predisposing SNPs. So this is what happens during development. The red bubbles would start appearing, but actually would not convert into some phenotypic pathological phenotypes immediately. It has to reach some critical mass, and only at that point we start seeing psychotic symptoms. So that was our understanding of complex non-Mendelian disease about 5 years ago. Now, another question was like directly related question. Why is it so that, why and how DNA is aging epigenetically? And some completely unexpected insights came from another study in the lab. And actually now when I think back, we started investigating cyclical processes, cyclical epigenetic processes. And it's hard to explain actually rationally why we did so. Like we detected some evidence for intra-individual variation. We could not understand what is happening. And there was this weird idea actually that maybe some epigenetic processes cyclical. And some ideas came from circadian biology. So circadian oscillations is a very universal kind of phenomenon. And as you probably know what I'm talking. So the Earth is turning around its axis, like if you can see it. And then actually, it's been like this for 4 billion years. And life, even primitive forms of life, kind of started reacting to this kind of regular change of day and night. And it's easy to understand. So during the day, it's nice and warm, and it's a good time to perform various biochemical reactions and to synthesize whatever proteins. And at night, it's dark, there is no UV, or UV level is much lower, and actually it's good to replicate DNA because there are no mutagenic factors, or the number of such factors is reduced. So what happened that actually this kind of environmental, purely environmental factor at some point got internalized. And cells and tissues and organisms internalized this 24-hour clock. So it became part of the molecular processes, and it started regulating various biological phenomena in the cell. So the clock, which is basically was imposed from outside, became part of biological machinery. And there are so many different things actually that have been investigated from the circadian perspective. And we know that gene transcription oscillates in a circadian fashion, and proteins oscillate in a circadian fashion, and metabolism is circadian, and some aspects actually of epigenetic regulation, more specifically histone modifications, oscillate in a circadian fashion. So we thought, okay, like despite the fact that our understanding about DNA modification is something very stable, like written in stone, and I mean one cells kind of reach the final differentiation stage. They're not stable until they reach that stage. During development and differentiation, epigenomes are changing very dynamically. But actually, once they reach that stage, the understanding, the textbook rule would be that actually epigenomes are stable. DNA modification profiles are stable, and they may change gradually a little bit, like if aging here and there could be some stochastic processes, but in principle, they're stable. So we questioned this, we challenged this idea, and we performed some circadian DNA modification studies. And here is the design. So we did two groups of studies. We investigated animals, mice, and we investigated humans. And so the first thing is related to epigenetic aging, and that was done in the mice experiment. And here is the design. So we had three groups of animals of different ages. So different ages enabled us to investigate linear aging processes, linear aging, epigenetic aging dynamics. Three groups. And in each group, we sacrificed 2 to 3 animals every 2 hours in order to investigate circadian oscillatory processes. So we had 2 dimensions, so cycles and linear changes, circadian cycles, circadian oscillations, and linear age-dependent epigenetic changes. From technical point of view, we used a technique called padlock bisulfite sequencing, which is very informative technique, very flexible. You can investigate whatever targets you like. You're not attached, let's say, to Illumina target list, but for whatever reason, it's not frequently used. So I would kind of recommend to use this technique wider. And the key point, okay, so this is like one technical data slide, which is very busy, but the essence is that actually we did see epigenetic oscillations in some cytosines. So these are kind of extreme examples where those oscillations are very evident, but basically like as expected in a circadian kind of context, DNA modification goes up and then goes down and goes up again and so on and so forth. And this cycle is very close to 24 hours. So these are some examples. So not all cytosines oscillate. There are some specific group. We still don't understand why some cytosines oscillate and some others do not. But we see that actually, let's say there are some genomic elements exhibit different densities of oscillating and non-oscillating cytosine, but that's a separate topic. I'll not go into that today. So in terms of like, what's the connection between cyclical processes and linear epigenetic aging? As I mentioned, we can investigate modification dynamics in 2 dimensions. So we have oscillating modified cytosines in each group that we could interrogate in those samples collected every 2 hours. And we have this vertical age-dependent epigenetic changes. And we asked a simple question. So we generated 2 lists of cytosines, the ones which oscillate in a circadian fashion and the list of cytosines which show linear age-dependent epigenetic changes. And the question that we asked, would the 2 lists overlap more often than we would expect by chance only? This was the key question, actually, like, would they overlap more often? And yes, they do overlap more often than you would expect by chance. So in both tissues that we investigate, liver and lung, we generated statistically significant evidence for statistically significant overlap. So it means that the 2 processes are connected, the 2 processes are related. So a cytosine is oscillating, there is a chance that actually it will also exhibit those slow age-dependent changes. So our interpretation is that actually, like, once we see this regression to the mean, like a typical feature of epigenetic aging, so we start with some kind of extreme low density of modified cytosines, high density of modified cytosines, and they regress to the mean. And they not just regress in some chaotic whatever fashion, but actually they are cycling, first of all, and they also have this linearity, some linear trend, which would basically, we would eventually register as age-dependent epigenetic changes. So linear effects and cyclical effects seem to be connected. So now we want to kind of go back to that initial question, like why is it so that actually different haplotypes would exhibit differential epigenetic aging, and what would account for that? And the revised model is now that actually it's not that they are gradually changing, but actually those cytosines which exhibit age-dependent changes, they would also exhibit oscillations earlier in life. So that is our revised model. So effectively, like the stuff that we are working on now is like we want to investigate kind of genome-wide effects and test how different polymorphisms and different haplotypes would explain, would account for differential epigenetic aging. So this question is directly related. I have another slide, this one more complex, and you can see we have three groups of animals, two groups of inbred animals marked by blue color and pink, and we have the hybrid group cohort of animals, and then purple, and we can see that modification is different, and actually in some cases we see evidence for oscillation. So some haplotypes seem to be oscillating in an epigenetic fashion, and the alternative haplotypes are not oscillating. And if we investigate, let's say these are animals that are 100 days old, and these animals are 300 days old, and you can see that actually this whatever methylation QTL effect is changing. So that is another kind of point that MQTL could be also age dependent. And the whole idea is actually to connect like all these important facets of phenotype, genetic background, epigenetic dynamics, and the temporal dimension. So the way we understand now actually the entire epigenetic panorama is the following way that there are, so I was talking about circadian oscillations, but actually it's not the only one type of oscillations that is present, and there are various other kinds of oscillations that should be investigated, and they overlap and kind of synergize or antagonize each other. So there are ultradian oscillations which are very short. There are circadian ones that I talked. There are infradian that are longer than circadians, but not very long. Circalunal, depending, which basically would be consistent with the lunar cycle. And there are circannual. Circannual cycles would take full year. So basically, this is the way we imagine actually how those epigenetic systems, and not only epigenetic, like all biological systems in the cell are organized. They are subjected to various kinds of oscillations. And those oscillations interact and accompany us from birth to, not from birth, but actually from early developmental stages till the very end. And the idea is that actually disease is nothing else but disintegration of the system. And you can start seeing, and you see like all those gaps appearing. And basically at some point, this nice evolutionary important system starts collapsing. And that is the putative disease mechanism. So I also mentioned that we performed a human study, and we investigated neutrophils in this case, and I'm showing this in order to demonstrate the connection to disease. So the principle was very similar. We collected blood samples. We used sorting and separation of neutrophils. We purified neutrophils in this way. And we interrogated on, I guess it was the Illumina array at that time. And we also detected some fraction of cytosines exhibit oscillations. And then we did a kind of analysis of various kinds of EWOS studies. And so we basically, the only one kind of common denominator was that we used the same platform, but different EWOS studies were performed in different places of the world by different groups. and researchers had very different goals. And we investigated circadian oscillations, which basically these two studies were completely unrelated. Again, the only one common denominator was that we use the same platforms. And we asked the same question, like would the EWAS hits overlap more often with the oscillating modified cytosines that we identified in our study. So we had like, I don't know, half a dozen or so phenotypes. And as a rule, we did see statistically significant overlap between EWAS hits, which were produced completely independently from what we were doing in our lab, and oscillating modified cytosines. So that makes some interesting link, not only to aging, but also to disease, to the epigenetic basis of disease. So now our understanding, we revise this model, and actually we believe that in addition, like that those epigenetic changes are not necessarily kind of linear and gradual, but the oscillatory component is also very important. So we believe that actually those predisposing factors should be oscillating, and probably oscillate in the wrong way, in the wrong places. And that is actually what we expect in individuals who are to develop disease. So I just want to kind of make some comments. Let's say we would advertise. Let's say we would argue that circadian analysis, or let's say investigation of cycles, of epigenetic cycles, would be of interest, and in comparison to cross-sectional studies, such cyclical studies would be more informative. So here is an example. So if we collect like if we do some measurements in the usual way, and we have control group and we have patient group, and we see some differences, there are some difference in the mean and some difference in the variance. And this is what happens in traditional EWAS. So there are some small mean differences. Some studies detected variance, higher variance in patients, which is hard to explain. Where is it coming from? And basically that's usually the end of the story. So we are saying actually that if we would spread those dots in the temporal dimension, so usually samples are collected during business hours between 9 and 5. So we can start seeing there's some distribution of the dots, which is still not very informative, but actually those dots could be hiding the curves, the oscillation curves. And this is like ideal case scenario, an illustration what the controls would look like, and they're very consistent. They oscillate in a highly synchronized fashion. It probably does not happen this way in real life, but in the patients, we expect to see much larger variation. So some patients actually could be oscillating in a very similar fashion to the controls, and that specific spot, that specific region would not be a risk factor. But there are some other patients would exhibit major difference. They either flat, for example, they do not oscillate, or they oscillate in anti-phase. So it's like an inverted oscillation. So in those cases, we can suspect that actually we have an epigenetic risk factor of cyclical fashion, cyclical origin. And the full study would look like this. So if we spend enough energy and resources and we collect the samples at some specific time intervals, let's say every 2 or 3 or 4 hours, we will start seeing the curves of oscillations, and we can fill up the gaps, and actually in the picture, this entire situation becomes very clear. So this is our expectation. This is what we hope to see in disease. And that's why we advertise and we advocate for I don't know what to call them, longitudinal or extended collections, like multiple collections over some specific time intervals, which would exhibit, which would reveal some intra-individual epigenetic variation. So one of the concepts actually that I kind of did not mention before, but this intra-individual epigenetic variation is of significant interest, and basically like that has been neglected, ignored, and not investigated. So intra-individual epigenetic variation is a broad concept, and all those cyclical things represent part of intra-individual epigenetic variation. As I mentioned before, there could be multiple cycles, and those profiles actually in some cases are not easy to dissect. It's not easy to understand the origin and how many different cycles interact and overlap and co-act together. It's not a very simple task, but actually it could be quite informative and helpful. Yeah, so like what I described in terms of like all those epigenetic investigations is like only one layer, but the same principles can be applied in various other kinds of studies. And let's say we launched a pilot study using MRI, magnetic resonance imaging, and human brains. And we scan every individual every 3 hours. So we have like 8 or 9 scans in 24 hours. And we do see that actually various MRI modalities exhibit oscillations. It's not a static thing. So the MRI field, I'm kind of completely new to this field, but actually the MRI community is always complaining about inconsistencies of the findings, small differences between the groups and patients, and some kind of lack of stability of the signals, and so on and so forth. And circadian oscillations and other kinds of oscillations, cyclical oscillations, could be a fact actually which contributes to all the uncertainty. So the problems that they are facing are very similar to what we are dealing with in epigenetic studies of disease. So this is one example, and here's another example also showing oscillatory effects of MRI modalities in a small sample size, a pilot study, but we do see those effects very clearly. It's another example. So this is actually, I'm pretty much done. It looks like I was talking very fast. But actually, this is the group and the people who performed this work. And I went to note Gabriel O. and Sasha Brahimi and Matt Carlucci, who led the projects, the molecular epigenomic study, and that analysis was performed by my compatriots in Lithuania, Karlis Koncevičius, and this guy, Algis Kriščiūnas. And basically, that's probably it for now. So I have this slide. I just noticed that. So there was an article in a Time magazine in probably about 2010. So it's a Time magazine, and the title, "Why Your DNA Isn't Your Destiny." And it was a long and pretty comprehensive, pretty interesting paper about epigenetics. But the message actually here is subliminal. It's the time dimension in epigenetics that was not discussed in that article, but in my opinion is very important and could be very productive if we do it right. So that's probably it for now, and I would be happy to discuss and whatever you want to discuss and answer your questions.
That's great. Thanks very much, Art. So if people have questions, if they put their hands up, then Marie can unmute you, or if you're if you want to type them into the question and answer box, then you can do that. But yeah, if you put your hands up, Marie should be able to see and unmute you. Yeah, the simple, well, probably a naive question is, do you think you could learn anything from just conventionally measured, samples with methylation arrays if you knew what time of day they were taken? Do you expect to see anything, any signal? Because if you've had samples taken from early in the morning to end late afternoon, early evening, would you expect to be able to see any sort of time of day effect?
We tried, and we failed. And I think that the reason actually that humans are quite heterogeneous and basically are all those things, starting with chronotype, which is relatively easy to register. So chronotype means like would be early chronotype and late chronotype, depending on what time people go to bed and wake up. Yeah. So chronotype could shift things by 4 or 5 hours very easily. Yeah. Then actually there are numerous other things like age effects, like sex effects, and basically there are all those kind of genetic background effects. And we were not able to connect the dots actually. Like if we have only one measurement from one individual, like we were not able to identify oscillations, even if we knew, let's say, the time of death, which is basically kind of can be pretty precisely recorded, or basically the time of sample collection, we were not able to detect oscillation effects, no. If we have, like there are some studies, other studies where basically we have, let's say 4 samples per individual, then things become much more informative. Then actually you can kind of, adjust those things and do see the effect. But like from one single snapshot, you cannot restore, let's say, this kind of populational thing for various reasons.
So do you think that the patterns are sort of just completely individual, or do you think the patterns, show some, some coherent sort of diurnal?
So it's both actually. It's both. Now we investigate, like we have more data, and we do see that actually those epigenetically variable sites. I have to explain actually how we do this. So first of all, out of this big entity of cytosines, we identify cytosines which exhibit intra-individual variation. So intra-individual variation is not necessarily oscillation. Like intra-individual variation could be various things, yeah? So, but we work with this group of cytosines which exhibit intra-individual epigenetic variation. And fraction of that could be 10% or 20% or 15% or whatever, could be oscillatory, right? So we identify those epigenetically variable cytosines in multiple individuals, and we do see an overlap. But there are also individual specific sites which would be present only in that individual and not other individuals, which are variable. And we don't know why, yeah? So we're kind of still at this kind of descriptive phase where basically we're trying to understand, to what extent different people are different or similar in terms of this type of variation, not inter-individual, but intra-individual variation. So there are overlaps, there are similarities, there are differences.
Yeah. So Caroline has a question. I think she can actually speak.
Yeah, hi Art, thanks very much.
Hi Caroline.
Right, nice to see you.
I cannot see you, but I can hear you.
Yeah, thanks for a really great talk. I was just intrigued by your initial comment around the latency of complex disease phenotypes and this notion that you have a genetic lesion, but it doesn't really manifest itself as a disease till later in the life course. That resonated for me with the developmental origins of adult disease hypothesis, where the early life insult is environmental, but the same latency is experienced. I just wondered whether what your thoughts were on the parallels with your between your sort of chronoepigenetic hypothesis and the developmental origins hypothesis.
Yeah, so developmental origin of disease, like you have to refresh my memory. So idea is that actually there are some early developmental processes which have an impact on late life health. Is that correct?
That's correct. Yeah. So they could be in utero, or they could be early life.
Yeah. So the point is actually to understand what happens in early development, and basically like all those parameters like weight and obesity and so on and so forth, or undernutrition, how it would affect like low weight, for example, or overweight, how it would affect, let's say, whatever, like hypertension, atherosclerosis, predisposition to cancer, and so on and so forth, So I think that actually observation is very interesting. I want to make several points about this entire business. So the first point is, I'm trying to address your question. I think the observation is empirical, and basically we cannot deny it, like it's there. right? The question is like, what is happening? Like, how come those very early life events would contribute to something what would happen, let's say, over years, like over decades, in decades, yeah?
Yeah.
Like what kind of, how can we connect those things? Like what is going wrong here that would generate some bad outcome, years later. Yeah. So we need simply the point is we need working hypothesis. We need some testable theories or testable. We need to perform experiments, actually concrete experiments that would help us to address this question. Yeah. So go ahead.
So there's obviously been a lot of interest in epigenetics as an explanation for that latency as seen that you see in developmental origins, but certainly what we've seen, and it's been corroborated, I think, by other studies, is that you get changes or differences in early life in DNA methylation patterns, but they don't persist through the life course in a way that would explain that latency. And I'm just wondering whether there might be a temporal component to that that we're missing.
Yeah, so I think you're talking about the general problem, like what we're dealing with traditional epigenome, let's say epigenomic studies of whatever phenotypes, that the effects are very small. Let's say if we talk about DNA modification studies, we identify, let's say, differences, even if they are statistically significant, but they are small, as I said, and they are in some kind of in the middle of nowhere, like in some intergenic regions. And we have, let's say, we scan 850,000 cytosines, and out of this large number of cytosines, we have, let's say, 20 cytosines which would exhibit statistically significant differences between cases and controls. I'm saying, by all what I tried to explain today, I'm saying that we could be missing some major effects if we do this kind of snapshot type approach. If we apply the snapshot type approach, we may be missing some very important aspects of variation, of epigenetic variation, which could be quite informative. Yeah. I did not want to go into details, actually technicalities and so on and so forth, but if you look at that paper on chronoepigenetics that George mentioned, actually, there are some simulation studies showing, how can you miss some very major effects? Like, you could be anti-phasic oscillations and you may miss them. Mm-hmm. Yeah. Yeah. So I am saying that actually, like, if we do, this, like if we add the component of intra-individual variation, we may be detecting some very interesting effects, which eventually could help us to connect actually to some long-term outcomes. I don't know if that is the case, but this is a testable thing.
Thanks.
We may be missing something really important, and we're grabbing things which are basically like, by some fluke reveals some statistically significant differences, but biological interpretation is never clear.
Yeah, I wonder whether there are any approaches that have been adopted in the measurement of other factors that show, like cortisol or factors that show well-established diurnal variation.
Yeah, so there are a group of factors, actually, like everything is changing when you think about, like, what we do during the day, what we do during at night. Yeah, like there are so many different things that will change, like, starting with cortisol, melatonin, and so on and so forth. Like everything is changing, like, your blood pressure is changing, like there's some dynamics, like, cell count differences in the blood, like the proportion of neutrophils to lymphocytes will be changing. Everything is changing, yeah? So the question is, like, it all depends on what questions you're trying to address. Like, if you're interested in epigenetic basis of developmental origin of late onset disease, yeah, like this kind of, you can measure cortisol, but it's so non-specific, it's so, universal, that I'm not sure that actually you can connect to whatever phenotype, yeah?
Yeah.
Again, like if you're interested in, if you believe that actually epigenetic factors play a role, and this is the thing that actually, like, we can detect before the origin of disease phenotype, yeah? Like, before we start seeing the symptoms, like we predict that actually we will be seeing some different signature of oscillations. So we'll see some anomalies, like, we have oscillations we're not supposed to be. Some oscillations we're supposed to be in the norm are not present in individuals who are predisposed to disease. Yeah. So those things are already kind of, boiling, slowly, gradually accumulating. And that critical mass of epigenetic misregulation could take years and decades, and then things happening that we see in the clinic. Yeah, but we can already register them. Like if we investigate pre-symptomatic individuals, let's say high risk, ideal case scenario would be we take high risk individuals and we follow them up and we investigate their whatever intra-individual epigenetic dynamics before they develop disease symptoms.
Yeah, so I guess the difference between the sort of conventional developmental origins hypothesis and the chronoepigenetic hypothesis that you're positing is that the developmental origins hypothesis suggests that there's an epigenetic change in early life and it perpetuates, whereas you're suggesting that there's an accrual of that epigenetic change over time that then triggers the disease. So it's just, it's sort of slightly looking at it through a slightly different lens. But interesting.
I don't think there's major contradiction here. Like
no.
So John Mill has a couple of questions. If Marie unmutes John, then he can from all the way from Exeter. The people have digitally travelled for miles. So can you unmute John? Marie? Maybe not.
Doing it.
Yeah. So just while John's being unmuted, Art, I hadn't told you in advance, but you're not allowed to use the term statistical significance in Bristol.
What's a substitute?
It's banned.
Anyway, John, what's a substitute for that? You say the groups are different. That's it.
Well, you get the estimate of the group difference, and then the precision, and then your precision around the estimates of the group difference.
So, and it doesn't matter what p-value you have. Like, still, you cannot say.
No, the p-value will relate to the will relate to.
Okay, George, you'll have to educate me. You'll have to educate me in this.
I'll do that, but I'm going to read out John's questions. So, so John says, can you rule out that the effect you see in tissues, e.g. blood, etc., are not simply driven by circadian shifts in cell type proportions, which may be a different DNA methylation signature at a specific site, e.g. shifts in immune cells across the day?
Yeah, so we had very hard time actually responding to these questions. It was one of the major criticism of our reviewers for one of our papers. And yes, like we can do the best we can. So that was the reason actually why we separated neutrophils, let's say, or now we're doing T cells and monocytes. So, we use those cocktails of antigens, and actually then we test them on the sorter, and basically once we have epigenomic profiles, we use the Hausman algorithm, which we tell that actually like all cells are neutrophils, like we don't have any contamination of any other cell. Yeah, so we do this due diligence, we go as far as we can. The problem is that actually within neutrophils, there are subtypes of cells, and it could be young neutrophils and old neutrophils. And the difference between the two are not qualitative, they are quantitative. Yeah, so it is a little bit more of this antigen here and a little bit less of that antigen here, and vice versa. And yes, cell heterogeneity and circadian oscillations in cell counts could confound, could contribute to, could simulate epigenetic differences, could simulate circadian oscillatory effects in epigenomes. Yes, that's true. We do our best, like we try different scenarios, we investigate enterocytes, we try to come up with as clean population of cells as possible. But yes, that is a confounding factor.
And
Yeah,
a second question is from John is also, if you lock yourself in a dark room or fly across the world on a plane, do you envisage these circadian patterns change? And do they change across the year?
Would you expect some changes in a dark Lithuanian winter night?
Yeah, there you go.
They're not dark, actually, they're dark in the UK, in fact. The point is that actually, so this is the thing that actually like we have, like our cells, like talking about humans or mice or whatever, we have internal clocks. So it doesn't matter, like if we put, let's say, if we entrain an animal, let's say a mouse, yeah, in this whatever, 12 hours of light, 12 hours of dark, and this animal works like a clock, and then actually we put it in full dark for 48 hours, the animal would still have the clock. It would not disappear. It may start drifting a little bit, but actually the cells still have the clock. The cells still count, the cells still predict the time of the day or night. The cells still tell the body that now the time is to eat, or now is the time to sleep. Yeah, so those things, so this is the point that actually I'm trying to emphasize that this kind of, the most universal environmental factor, which is related to the fact that the Earth is rotating around its axis, yeah? And the day is changing night and vice versa. It got internalized, and it probably happened like billions of years ago. Like the first most primitive forms of life started kind of reacting to this, and at some point realized that probably it's better to have it inside. So it's a major evolutionary advantage, and probably that was so critical, important, and eventually, like, beyond what we're discussing today, those mechanisms probably have been hijacked and used for differentiation and development and so on and so forth, and building complex multicellular organisms. But that's a separate topic. But yes, like we have internal clocks, and actually internal clock is trying to adjust and kind of be consistent with external clock. And this happens, this adjustment is happening every day. So no need to blame dark Lithuania nights.
That's great. I'm afraid our hour is up. But we're going to join on another Zoom link. So you have to go into the second.
Oh, there are still some questions, George.
Yeah, but I think we're timed out. But the people who've asked them in the questions, they're going to be on the call on the. Okay. Right.
So we're reconnecting on this other link. Okay, great. Okay.
Yeah. So thanks very much. You can imagine.
My pleasure. Absolutely.
Imagine the applause.
Actually, it's very dark to talk. Like I just realized that I'm talking to myself and I don't see the sound. Yeah. Anyways, okay. So we'll reconnecting soon. Yeah.
See you on the next link.
See you.